Akshit's going to be talking to us about geometric approaches to predicting ecosystem responses to perturbations. And going on the abstract, I believe there's going to be consumer resource models. I believe there's going to be environmental variability and microbes. So yeah, I'm super excited. And yeah, I'll hand over to Akshit.
Perfect. Thanks a lot, Andrew, for the introduction. And yeah, you basically guessed just right. Those are exactly the three themes that I'm going to be talking about today. All right. So let me get started. And while I start, also maybe I'll try and make clear again what Andrew said, that I'm very happy to take questions in the sort of middle of the talk. So if you have any questions, please feel free to unmute yourself and just speak up, and I will stop and listen to your question. Okay, so hi, so I'm going to be talking to you about the geometry of ecosystem responses to environmental perturbations. And let me just begin by just reminding us what I feel are some central questions in population biology, right? And I would say really there are three key things that we like that many of us are often thinking about, and that they're all sort of interlinked. So of course, a lot of us would care about how diverse different populations and ecosystems are, how these communities assemble over time, how they might evolve over time. We're also, in principle, interested in how ecosystems and populations together provide some sort of function or perform functions, for instance, biogeochemical cycles that operate on Earth. And of course, also, we are very interested in what happens as things change, in particular, how do populations respond when there are perturbations? And of course, these three questions are not separate from each other. They're intrinsically interlinked. So of course, how ecosystems function is intricately related to how diverse they are, and how they respond to perturbations will change how they evolve over time. But I sort of want to spend the sort of majority of today's talk in this sort of bottom right part of this question pyramid, so to speak, which is the how populations respond to perturbations. And of course, this is a question that people have thought about for 50 to 60 years now. And I would say really the traditional focus when we've been thinking about responses to perturbations is responses to species perturbations. So let me describe what I mean. What we're typically thinking about is some ecosystem. Here in this cartoon, I've depicted a microbial ecosystem, but it doesn't need to be that. And so the key idea is that we're kind of imagining that this ecosystem is at some sort of steady state. And what we are interested in is asking what happens to this ecosystem when we perturb some or all of the species, and in particular their abundances, away from their steady state. And typically the kind of way that we ask this question is that we ask, the response is really to ask whether these perturbations will make it so that the system is stable, in which case you'll come back and the perturbations will die out, or unstable, in which case the perturbations will grow over time and the system could grow to either a different steady state or collapse or something else. Right? And this is, of course, an approach that was pioneered by Robert May way back in this seminal 1972 paper. And really, I would say it taught us this important lesson that's really served us well, which is that, you know, understanding response to perturbations, and in particular linear response to sort of perturbations, which is the mathematical way that we deal with this, is really a key way to understand complex systems, for instance, complex populations or ecosystems. And also sort of theoretically, it's given us grounding in that I would say, now have a pretty general framework for talking about stability of this kind. And that's sort of rooted in ideas from random matrix theory, but also in general from linear algebra and so on and so forth. These are the kind of mathematics that we use. But as well as we understand how ecosystems respond to species perturbations, we of course also know that these are not the only kind of perturbations that are going on there in ecosystems, right? There are many other perturbations that also shape ecosystems. And I would say there are two broad classes of these. So first, of course, is a class that I would say are abiotic perturbations. And so by this, I mean, if you have if you have some sort of ecosystem with some species, let's say these microbial species, and some resources that are being supplied at some rates k, and these species are eating the resources, maybe they're producing the resources, and these species are also dying. What could happen over time is that you could either have perturbations in the environment, which manifest as changes to how resources are being supplied, Or you could have perturbations of other kinds. You could have perturbations to temperature. You could have perturbations to pH. You can have perturbations to salinity. And what we typically imagine is that, well, maybe they change how these particular species are consuming and impacting resources or growing or something else. Right. So, of course, these are a class of perturbations that are a little bit distinct from just perturbing the species directly. But of course, there are also biotic perturbations, right? So in principle, you can also think of an ecosystem at steady state and some sort of invasion process where another species from somewhere else is sort of coming in and perturbing the ecosystem, right? So you can even think of invaders in principle as biotic perturbations. And just to sort of extend that further, even the process of how communities evolve in principle can be thought of this way, right? The only difference is that the invaders, so to speak, are not coming from outside the community, but they're coming from inside the community. They're mutants in principle of one of these species that are present in the community, right? So there's this broad class of perturbations that shape ecosystems, but like we have a general framework for dealing with species perturbations, I would say that we have lots of work on dealing with specific perturbations that's been a little bit hard to generalize. So just to give you an example of this, many people, including I'm assuming people here, have worked on responses of ecosystems or species to changes in temperature. Right? And sort of the hallmark way to think about this is in terms of these things that are called temperature performance curves. Right? This is just an example of this. And the idea is that the relative performance of some species, for instance, the growth rate or something like that, changes as a function of temperature, right? And in particular, there's some range of temperatures that sort of correspond to where species grow. So they can be cold water fish, warm water fish, and so on and so forth. And of course, there's been a lot of work that's been done on understanding how changes in temperature would manifest in changes to how these populations respond. But I would argue that just because we understand responses to temperature doesn't mean that we have a general framework for responses to all sorts of environmental perturbations, right? And the reason is that the physical mechanisms that elicit responses to temperature we expect to be fundamentally different from responses to other things, right? For instance, to changes in resources, to changes in pH, and so on and so forth. And so it's because of these differences in physical mechanisms that our work that work on specific perturbations seems hard to generalize. And so what that leaves us is that we still don't have the sort of general framework to answer the question of how ecosystems respond to environmental perturbations. And of course, this is something we should care about, right? This is something that has both practical importance as well as theoretical importance. Practically, of course, many of the questions that we would like to care about, especially, in a changing world with all sorts of environmental changes that are happening, we would like to know what happens, and we would like the ability to be able to predict how ecosystems would change if we change, for instance, some resources, or if we have some runoff of some new nitrate into some community. What new state will that ecosystem go to? And of course, like I already argued, we've learned from work by May and others in species perturbations is that actually the responses to perturbations seems to be a key way to just understand and characterize systems themselves. This is, of course, pioneered, the way that we think about systems in physics is exactly just poke at a system, see how it responds, and that tells you a lot about how the system behaves. Right? And so this is kind of, I would say, a missing gap that I'm going to try and address in this talk. And so just to sort of orient you to where we are headed, really my talk is going to be to show our progress towards building this kind of general framework for how ecosystems respond to perturbations. And the idea here is going to be that I'm going to tell you about a general framework that can predict how a community of different species will shift or change if I make a perturbation to the environment. And really the idea is going to be this framework is general because the models of the communities that we're considering could in principle be general and arbitrary, but also the class of perturbations that we're considering could in principle also be general and arbitrary. And so what I'm going to show you after I introduce this framework is that not only can we make predictions about how communities shift, but also I'm going to show you that this kind of framework has a sort of a really a beautiful geometric structure that not only is sort of mathematically very nice, but also provides very interesting biological insights. And so one of those insights that I hope to share with you later in this talk, is that it can explain why, for instance, there are many cases where even completely cooperative communities, for instance, that are held together by nutrient exchange, can often behave exactly like competitive communities, meaning that if you're actually using ecosystem responses to perturbation to kind of figure out whether a community is cooperative or competitive, you actually will fail. And that I think actually has some pretty interesting implications, which I'll hope to dig into towards the end of my talk. So that's kind of the agenda for the rest of my talk. So let me sort of begin. So I told you that I want to talk about a general framework, but we've got to start somewhere to sort of root ourselves. And so where I would like to start is in this sort of canonical model of sort of ecosystems, which is a consumer resource model. And so we'll see that in this consumer resource model, it's going to be easy for us to think about how to perturb the environment. And so just to sort of introduce you to what I mean by these kinds of models, really in these models, we have a set of species or consumers, and we have a set of resources, which, are these arbitrary triangles over here. And the idea is that these resources are supplied at some rates, K. These species consume these resources, as a result, depleting them. And as these species grow, they face some mortality or maintenance in the environment, which sort of creates this nice balance between consuming resources and staying alive. Right. And so really to fully characterize this model, you need two sets of differential equations. One is a set of equations that tell you about how species abundances change over time. That's n sub I. N is the abundance of the species. I, the sort of index I is going to mean species I. And so really all this model is saying is that each species will grow by consuming different resources with some consumption coefficient Ci alpha. Of course, that consumption is going to depend on how much resources are alpha are available, and then they'll face some mortality because of which they might die. Right. And then the resources are also going to have similar dynamics in that in the absence of any consumers, these resources are going to be supplied, they're going to grow and then settle to some sort of carrying capacity. But then if you provide consumers, if there are species available, then these resources will get depleted. Right. In principle, of course, we can talk about more complex consumer resource models, which I'll talk about later in my talk. But for now, this is sort of enough to get us started. And just to orient ourselves, the environmental parameters that we're going to think about here are these two things. We're going to think about the supply rates of different resources, K alpha, and the mortality rates, or how harsh the environment is in terms of MI. And of course, when we're going to ask about the response of different things in the ecosystem, we're of course going to care about how species abundances change and how, in principle, also resource abundances change. Right? So those are our response variables. And so, of course, what we'll be interested in asking is if I make a change delta M to the mortality in the environment and a change delta K to the supply rates, these are my environmental perturbations. How am I going to see a response? How much am I going to change the resource abundances and, in principle, also the species abundances? Right? And if you just do out the math for this, really the math's not that important. It turns out that simple calculus will tell you that the key quantities that you need to worry about are these sort of derivatives here, right? These are really what in physics language are called susceptibilities. In other languages, they're just how ecosystems respond, right? So many people also call them what are called response functions, right? It's literally just asking, if I make a small change to, for instance, mortality, how much does the species abundance change, right? And so the key task here is really to compute what these susceptibilities are. If we can compute these four susceptibilities, then we've in principle computed the response to the environmental perturbation. So it turns out that in the MacArthur consumer resource model, this is actually straightforward to do. You just need to do some sort of linear algebra, and out pops out these four forms of these susceptibilities, which are in terms of some matrices. Right. And again, what these matrices are exactly are not so important at the moment, but I just want to kind of highlight that these matrices have very interesting ecological meaning. These matrices come out as emergent quantities of this kind of linear response. And so, of course, just to orient you here, these delta M's and delta K's are environment perturbations, and these are exactly the susceptibilities that we computed. And some of these quantities, for instance, the A is just the effective species-species interaction that you might be used to if you're thinking about a Lotka-Volterra model or any sort of pairwise ecological interaction model. right? And of course, these C's and E's are the consumption and impact matrices. And so really, if you kind of stare at these equations for a while, what you'll sort of realize is that not only does this sort of solve our problem in that it tells us how to predict responses in this MacArthur consumer resource model, it also has actually a very interesting mathematical and in fact geometric structure. So really the key idea here is that there are four quantities, right? There are, of course, species abundances, resource abundances that live in the space of species abundances and resource abundances. But in principle, there are also fluxes or rates, right? And in fact, there are two quantities, the mortality rates and the supply rates that live in a different space that have sort of dimensions or quantities that are not abundances, but they're sort of rates, they're sort of one over time. Right? And really what this equation is telling you is that it's telling you how to sort of transform a variable or a vector from one space, which is a rate space, to another space, which is an abundance space. Right? And it's really this quantity, these sort of susceptibilities are doing the job of mapping between different spaces. Right? And so really you can kind of formalize that picture in terms of this sort of cartoon that I like to draw, which is that you can really draw out what these four spaces look like. Right? So there are these four spaces, there are sort of species and resources, abundances and fluxes. And each of our four key quantities in our consumer resource models live in these four spaces. So our species sort of species abundances is a vector that lives in the space of species abundances. Species mortalities, in fact, live in the space of species fluxes, right? So these are actually rates. So this vector, which is a vector that tells you how much the species one die by, how much the species two die by, how much the species three die by, that actually lives in this different space, right? Similarly, resource abundances live in a resource abundance space, but the supply rates live in a resource flux space. Right? And really what our linear algebra is just telling us is that if you want to predict how species abundances will change if you make a change to the supply, what you really have to do is to ask yourself how you would transform this vector from this space in which it lives to a vector that lives in this space. And really, the key idea is that these susceptibility matrices that I was talking about in the previous slide are just doing the job of basically transformations, which is that, I've sort of drawn out this sort of network here. And so all this really means is that if I want to ask what is the impact of supply on species abundances, I have to take this circuitous route from supply to resource abundances to mortality rates all the way up to species abundances. And so really when I do this, I'll pick up these sort of transformations. I'll pick up, so this is just I, this is just one, but this over here is C, this is the consumption matrix. And this over here is the inverse of the species interaction matrix, right? So it's really by composing all of these operations that I'll end up going and asking myself, how does a change in supply really manifest to a change in species? And it's in fact this exact thing that you can see over in this equation over here, right? So just a delta K affects delta N because you take a route through C and A in right? And so that's really the way to understand and encapsulate all of these equations and in terms, I think, of this graph. Right? And so really these susceptibilities act as maps between vector spaces. And so actually this operation turns out to be very interesting because it turns out every time you take a vector from its native space to its non-native space, to any other space, you actually get a new emergent ecological quantity. Right, so this I've tried to sort of highlight in this sort of matrix over here. So the rows here kind of tell you what the original vectors are. And so this is, again, there are four classes. There are species abundances, species fluxes, resource abundances, resource fluxes. And then the columns over here tell you what space we can transform them to. Right, so this again would be the same set of vectors. And so again, we can see that if I take like a vector in the resource flux space, which is our external supply rates, and I want to transform that to species abundance space, I'll end up getting this quantity. And actually, each of these quantities has an ecological meaning. And those meanings are kind of what I've written out here. I don't want to go through all 16 of them, but just sort of highlight to you here what this quantity means. All this is just telling us is really how much abundances do species get if all they're doing is that the resources are available at some supply rate, right? There's no death, there's no nothing, just what is the naive abundance of these species just due to the supply rates. And similarly, you can sort of map out this entire zoo of these different transform vectors, which I think have this was a fun exercise to sort of come up with all these sort of emergent names. So this sort of geometry is mathematically interesting, but also I think ecologically interesting. And so now because of this, you can sort of describe these equations, not in terms of equations, but in terms of a geometry, right? So you can look at geometry in species space. You can also look at geometry in resource space. And so just to sort of tell you what this looks like in the next 1 minute or so, we have our species abundance vector that lives in a space of species abundances. And really what our math is telling us is that this vector is actually a sum of two different vectors, two different emergent ecological quantities. One is to do with supply and the other is to do with mortality. So the sort of pink here comes from what happens if you take the supply vector and transform it to the species abundance space. That gives us what the naive abundances would be due to the supply. The other vector is asking what happens if you take the mortality vector and transform it to the species abundance space. And what that tells us is sort of how much mortality burden these species are facing. And so really you can understand the steady state abundances of these species as a vector sum of just how much naive abundances you get from supply minus how much mortality you face, right? You can do the same thing for resources. So you can take, for instance, in resource supply space, what your resource supply is. It turns out that if you ask, you can sort of decompose this in two nice ways. There's an occupied net space. This is just all the resources that species could ever eat. And basically what happens is that you essentially what the geometry is telling you is that you project your resource supply onto this occupied net space. So species can, of course, only eat stuff that lies in what they can consume, but they won't consume all of it. So the steady state resource supply is not this vector, but in fact, they're going to face some mortality. And so really you have to take the mortality vector now and transform it to the resource supply vector. And really what you get is sort of two things. One is a set of unutilizable resources. That's the sort of dashed stuff here. And the other is the sort of utilized resources, that sort of stuff here. And really the steady state resource supply is again a vector sum of what is left over and unutilizable and whatever is utilized but not fully utilized because of mortality. Right. And so this is sort of the nice thing. You get this nice geometry. But of course, the point that I also want to make is that this theory can actually make accurate predictions. And of course, I wouldn't be coming to you telling you about all of this if the predictions that it made were not right. Right. So just to sort of orient ourselves and highlight here, this is like an example from a very simple two species, two resource version of this consumer resource model. I've taken some random parameters for growth and impact. And what I'm just going to do to test if the sort of theory makes accurate predictions, that I'm going to compare two solutions. One is that I'm going to simulate this model and run it to sort of steady state, no theory. And I'm going to solve for the steady state species abundances and resource abundances. And the second is I'm going to actually try to compute them using those formulas that I wrote over in the previous slides. And I'm going to compare them. So just to sort of orient ourselves, when you simulate stuff, you see that the species over time and the resources over time, they change, they have some transient, eventually enter some sort of steady state where both species survive, both resources survive. And I can represent that geometrically in this space of N1 and N2. This point over here is the N star, the steady state species abundance vector. This point, this vector over here is the R star. That's the steady state resource abundance vector. And if I know what the supply and the mortality are, I can also make a prediction of the same steady state vector from theory. And so that's those red, that's the red and the blue vectors over here. And you can see that basically they match very, very well. But not only do you get stuff matching very, very well, you can see that basically the steady state abundances come from that decomposition. You can sort of decompose the steady state abundances into two parts. One part that comes from just the supply, that's the pink thing over here. And the other part that comes from mortality. And so you can in fact see that the steady state abundances is the pink minus the blue, right? And of course, now you can predict what would happen if you change the mortality. If you change the mortality, you change how big the sort of blue vector was. And of course, then you'd concomitantly change how much steady state species abundance is. Right, so of course, this works in a simple setting. This is all about MacArthur consumer resource model, which is simple. You can do linear algebra and so on and so forth. But I want to sort of highlight here very quickly that actually this framework, this idea of thinking about susceptibilities as these key objects actually is universally predictive and applies to a broad class of models, and in fact also to a broad class of perturbations. So not just resource supply perturbations or mortality perturbations, but a very broad class of perturbations. And so I won't go through all of the math, but I just want to give you the key idea of what is sort of relevant here, which is that the key idea is that for any complex model that you might write down, there is a simple effective model near steady state. And that simple effective model near steady state actually looks exactly like a MacArthur consumer resource model. And that actually highlights why these kinds of consumer resource models that we write down that are admittedly very simple seem to work so well very often, especially when you make small perturbations. So the idea here is that now I'm going to consider a very general consumer resource model. And so and to show that it's general, my growth rate or my growth rate function is some arbitrary function gi. I don't know what it is. It could be anything. And for my resource abundance, for my resource dynamics, I could have some arbitrary supply function, some arbitrary resource interaction function, but also in principle, some arbitrary resource impact function, right? So this could be, for instance, a situation in which you have cross-feeding. You could have species consuming resources, but also producing resources, right? You just appropriately modify this F function. So you can have any arbitrary G and F. And the idea is, as long as you assume that you are near some known steady state and you want to predict the response to perturbations, all you need to do is sort of essentially linearize these models, and out will pop out a MacArthur consumer resource models with effective consumption and impact and species interaction matrices. And you can understand these effective matrices essentially as some calculus done on these arbitrary functions. Right? And of course, so now you can take any function in principle that you define and calculate these effective interactions. I won't go through doing that for you because that's just boring math. But the idea here again is that basically now instead of looking at now again, you can make environmental perturbations. You can go from some supply vector k to some supply vector k prime and really ask, if I change this supply by some delta k, and I change the mortality by some delta M, what is the response or what are the changes to my species abundances and resource abundances? And it turns out exactly the same formulas that I wrote down in the previous slides work out in this case because you're near some steady state and there are these effective interactions. Right? So this idea of this linear response should just work for any of these models as long as you know what these G's and F's are. But again, I sort of only pointed out to you that they work for if you change the supply or you change the mortality. And so again, so the sort of idea here is that it turns out, and we found out that even complex perturbations, for instance, changes to pH, changes to temperature, mathematically can be decomposed into effective perturbations to a supply and mortality. So I won't go through the math of that here, but the idea here again is that this sort of starts to build towards a more general framework because now you can take kind of any perturbation, do some math on it, and ask, okay, what is the effect of that perturbation if that weren't instead a perturbation to pH or temperature, but instead an equivalent perturbation to supply and mortality. And using that, basically, with this effective model and with these effective perturbations, the same math applies. And so let me just show you some examples of that kind of working out, because it turns out that this works out for community data. And so I'm going to show you quickly two examples. The first example is of pH sensitivity in denitrifying microbial communities. And so the idea here is that basically denitrification is this process by which basically communities collectively convert this important molecule nitrate to nitrite, to nitrous oxide, all the way to atmospheric nitrogen. And this, of course, is really important for the nitrogen cycle. And the idea here is that microbial communities perform this operation, but there are some microbes that can only perform nitrate to nitrite, some microbes that can only perform nitrate to something else, to nitrite to something else, and then some other microbes that can, in principle, are generalists that they can perform sort of both or many operations. So in principle, what you can do and what was shown in this paper is that you can essentially derive from experiments a very complicated looking consumer resource model, which is by no means a MacArthur consumer resource model for how these communities behave at different pHs. Right, so the idea here, again, I don't really want to flash and explain all the equations. The idea that I want you to take away is that it's not a MacArthur consumer resource model. It's a complicated model in which nitrate to nitrite and so on and so forth are being converted. And there's some nonlinear effect of pH and how it changes, for instance, the consumption rates and so on and so forth. And so that's all I want to do to sort of flash these equations. The main idea that I want to highlight is that the way that pH affects these communities is that basically nitrite becomes toxic as pH decreases. So the effect of nitrite is that is to have a pH dependent toxicity, basically. And so you can essentially now simulate this model. What was also done in this paper here from Sepekun's lab is that they did experiments at different pHs. And so what you can do is you can sort of change the pH and ask how these communities respond. And so you can do that in simulations. You can also do that in experiments. So the idea here is the following. You can start off, for instance, at a community at steady state at some neutral pH. Right, so if you simulate these dynamics with two species, N1 and N2, you'll find that their species abundance is settled to some steady state. And now what you want to do is, let's say you want to change the pH, right? And you can change the pH to say, let's say, reduce it by a small amount. And what this will do is, of course, it will elicit changes to the species abundances, which will now settle to some new steady state. Right? And so the idea here is that changing the pH was now our environmental perturbation. And the response that we care about are these changes to the species abundances. And you can go and do that for many, many simulations with many, many different parameters. And you can essentially see one very interesting trend, which is basically that generalists seem to be more sensitive than specialists to these pH perturbations. And the way to see it is the following. So here what I've plotted is the change in abundance of species 1 versus the change in abundance of species 2. These are just two species. And what I've assumed in these different simulations, each point over here is a different simulation with different parameters for this model. And the key difference between the blue and the red models is that in the blue models, both species, this sort of red and pink species, are generalists. That is, they can both eat nitrate and eat nitrite and convert them downstream. Whereas in the red, both species are specials. So the red species can only consume nitrate, and the orange species can just consume nitrite, right? And so what you can see is there's that across many, many same simulations, specialists are very, very symmetric and similar in how they respond to perturbations. That's why they sort of kind of lie on this x equals y line somewhere near the center. But generalists can be very, very different. And in particular, they show this very skewed sensitivity, right? So very often what you'll observe is one of the generalists has a large response to this perturbation, whereas the other generalist has a relatively small response to this perturbation. And of course, things can vary as you change parameters, but in general, this is true that specialists are all the way clustered over here. Generalists can have very, very differential sensitivity. And this is, in fact, also what is observed in these experiments from Sepekun's lab. Where they had lots of these different species, some of which are generalists, some of which are specialists, and they observe kind of the same trend, which you can capture with this model. But also this theory that we have here, this sort of geometric theory, explains why that might be the case. This is not necessarily something that you can do with the experiments, I hope you believe me. But what the theory is really telling us is that the differential sensitivity of generalists really lies in how the pH perturbations are decomposed into their effects on mortality and resource supply. And so the idea here is that basically I can take those same set of simulations on the previous slide and just talk about one of them. So here I've taken a case with generalists. I've again plotted the changes in abundance of species 1, the changes in abundance of species 2, and just asked how much they are for one particular simulation. That's the simulated vector. I can now decompose that using theory into, of course, the same vector that I can predict, but also in principle, I can decompose that into how much of the pH changes come from changes to the mortality to these species versus how much of that come from changes in the resource supply of these two species. And you can see that there's kind of this interesting effect, which is that the same change in pH decomposed into sort of reducing the mortality of both species roughly equally, right? So both these generalists have roughly a similar response. Really, the differential sensitivity comes from the fact that the supply is skewed toward one species, right? And so the sort of intuitive idea here is both of these generalist species are eating both of these resources, nitrate and nitrite. Both species kind of start to die, but just because of how things are, one species is sort of a little bit better, one is a little bit worse. And so because of that, the species that's worse starts dying, and all of the differential fruits of that come in as essentially extra resources that are coming to the slightly fitter generals. Right? And so the skew really comes from this, from how this pH is affecting the effective resource supply of one of the generals. You can do the same thing for specialists, and there actually you'll see that this same skewed effect of the resource supply is not present for generals, right? So the effect on mortality for both the specialists is roughly the same, but it is indeed true that the effect on the resource supply is kind of very, very similar as well. And so this is kind of what explains, we think, the differential sensitivity. And the key idea is really that the change in the pH has a different action on mortality and supply in generalists versus specialists. And it's really this change in supply that's really causing the difference. You can also take a second example. The previous example was about abiotic perturbations, like I was saying, that's changes to pH. You can also take an example of biotic perturbations, in particular, how communities shift when you invade with new species. This is the example that I was bringing up sort of earlier on in my talk, where you can kind of have a community at steady state. You can have an invader that comes in from outside, and the task is sort of twofold. You want to predict what the whether the invader is successful, and if successful, what the invader abundance would be in this new steady state community, and then how will all the communities community abundances shift. And so here, we again use sort of data from different kinds of communities. First, I'll show you stuff from plant communities, so not microbes, even though that's what the cartoon is showing here. So here is an example from this paper where they had different plant communities invaded by different species. And what we're trying to do here is on the y-axis are measurements made in these experiments about what the invader abundances were like and what the community abundances were like after this invasion, which is acting like an effective perturbation. And the x-axis over here is our prediction from our linear response framework. And you can kind of see that over orders of magnitude, we can predict both roughly what the invader abundances are going to be after this perturbation, as well as what the community abundances are going to be after the perturbation. We can do that not just for these plant communities, but we can do that for a whole host of different communities. This is an example of herbivore algae communities, where again, we're predicting the invader abundance and the community shifts. And you can see that kind of the predictions work well. And this is an example of microbial protists, these ciliated protist communities from this paper, where again, sort of the point kind of stands that this framework kind of does really well for arbitrarily complex models, well, in fact, even also for these realistic situations, right? So those are kind of two examples that I wanted to show you of the predictive power of this framework, right? Both for abiotic perturbations as well as for biotic perturbations. In the last part of my talk, I really just want to spend some time talking about the fact that this framework is not just predictive, but it also, I think, provides fundamental insights into how complex communities behave. And I just want to focus on one example over here, which is really this idea that when perturbed, cooperative communities actually behave indistinguishably from competitive communities. So maybe I'll tell you what we see first, and then I'll tell you what I think the implications are. So what we see first is pretty straightforward. You can sort of run a sort of model with microbial cross-feeding. This is a relatively complex model where there's one resource that's supplied, there's one species that can eat it, and then it produces a whole zoo of different metabolites via cross-feeding, which then other species in the ecosystem can eat. And what you can do is you can sort of assemble many, many of these communities at steady state. And for any one of those communities, perturb them around steady state by perturbing the supply of this nutrient and measure an effective species-species interaction matrix. Remember, this was one of the susceptibilities that we sort of cared about. And so this is actually one of the quantities that we can predict. And so I plotted that here, like one of the effective species interaction matrices for one of these examples. And just to orient you here, the rows and the columns are different species. They're indexed in some arbitrary fashion. And the color bar is such that all the competitive interactions, the effective competitive interactions are all in different shades of red, and all the effective cooperative interactions are in shades of blue. And I hope you'll agree with me that the way that I've constructed this model over here, this entire community exists in the first place because it's held together by cooperation, right? There's really a lot of cross-feeding going on, without which you'd really just have one species and no interactions to speak of, right? So the fact that you're getting interactions in the first place is because of the cooperative aspect of the community. And yet, when you actually look at these effective species interaction matrices, I don't see a single blue, right? You don't see any cooperation at all. So in some sense, when you perturb these communities, even these cooperative communities, wholly cooperative, behave exactly like competitive communities, right? And I think this is actually pretty important because there's been a lot of debate, especially I don't know about in the other literature, but especially in the microbial literature, about whether it is true that it's more common that you see competitive interactions or cooperative interactions, right? And there are lots of papers that people write about, oh, I think it's competitive interactions, and oh, I think it's cooperative interactions. And I would say essentially most of those papers are based in the idea of measuring these kinds of matrices, right? Maybe not the entire matrices, but subparts of these matrices. What you really do is you're looking at communities in different situations when they're perturbed and asking how species respond. And some people say, oh, well, I see competition, right? And there's this big narrative of how actually many people would like to say that, oh, it's all really competitive um, because, oh, look, these interaction matrices are competitive. And I would say that that is maybe a dangerous road to take, right? Because just this cartoon example, which is wholly a cartoon, shows that even the exact opposite regime, where there's full cooperation, it's still basically impossible to tell. You would make the wrong inference, right? And so, in fact, you can kind of play the same game. You can have the same interactions that you observe with a totally different mechanism. Now, I can take the same kind of species and put them in a completely different situation where I supply all of the resources so that they all essentially, this is an equivalent model, except you have a totally different mechanism of coexistence. They're all competing with each other. And I can measure the effective species interaction matrix here. And I hope you'll agree with me that basically qualitatively, these matrices are the same, right? They both look competitive, right? Of course, the matrices are, not exactly one for one the same, but qualitatively, they both show competitive interactions, even though one of them is bona fide held together by competition, the other one is held together completely by cooperation, right? And so it's really this kind of insight that, and then you can sort of go through all of this math of this linear response and kind of see why that might be the case. So I won't bore you with the math. I really think this message is, I think, an interesting and powerful one. And this is kind of what I'll leave you on. And so I'll just quickly sort of summarize. Sort of in this talk, we developed a general theory for how ecosystems respond to perturbations. By no means do we think that this is a complete theory, right? But it's sort of heading in, we think, in the right direction of generality. I think this theory kind of teaches us something important, which is these susceptibilities are sort of the key quantities that connect environmental perturbations to how ecosystems respond. And so in principle, these are the kind of quantities that one would like to measure. In particular, these susceptibilities also have this geometric interpretation as transformations between these distinct spaces. And it's really these spaces, I think, that are important to think about. And this geometry aids in the interpretation of these complex emergent quantities like niches and pH sensitivity and effective interactions. And I think it's trying to teach us this universal lesson, which is that if you look at responses to perturbations of communities, it's going to be nearly impossible to, for instance, tell what kind of mechanism is actually creating that community in the first place, right? And so with that, I'm very happy to take questions. Thank you so much for listening.
Many thanks, Akshit. That was wonderful. Really, really